Okay, we're going to go ahead and get started. Thank you all for joining us. I'm Young Lee. I'm the president and CEO of the Demon Runyon Cancer Research Foundation. And today we've invited a smaller group of our most engaged supporters to let you know how much we care about the work that's ongoing through our Damon Runyon scientists. We wanted to give you an opportunity to hear directly from us about how we're navigating this ongoing situation that has been so challenging for everyone. Since mid-March, we have been able to shift our operations remotely to protect the health and safety of our staff. And we've maintained our schedule of selection committee meetings for this spring by video conference, so our ability to give out new Damon Runyon Awards has not been impacted. Forging a career in cancer research is challenging under the best of times, and these are certainly not ideal circumstances for any of us. You'll hear about this in more detail from our scientists. Most of their labs closed down about 7 weeks ago, and these closures have been extremely disruptive to experiments in progress and have forced researchers to switch their focus to work that can be done outside of the lab. This time in the lab is especially valuable to scientists early in their careers as they are working to establish themselves in independent roles and become experts in their fields. When we select someone as a recipient of a Damon Runyon Award, we feel strongly that we're making an investment and a promise to them for the full duration of their award term, up to 4 or 5 years, depending on the specific award program. In order to see that promise through, we are announcing that we will extend funding for up to an additional 6 months for our Damon Runyon Fellows and physician scientists who are in the final year of their awards and in need of the extra time to complete their very important research projects. This will total an additional investment of up to $1.17 million this year. This additional investment is made thanks to the incredible support and leadership of our board of directors who have been crucial in helping us to navigate this uncharted territory. This extraordinarily challenging time presents an opportunity to live up to our mission and support young scientists in a meaningful way that makes it possible for them to successfully continue their careers in cancer research. I'm thrilled to have 3 of our current Damon Runyon scientists here with us for today's webinar. Please know that you can submit questions for them in the Q&A box on your screen in the bar at the bottom, and we will address as many of them as we can, time permitting. So I'd like to welcome Caroline Bartman, a Damon Runyon Fellow from Princeton University, Kyle Daniels, a Damon Runyon Fellow from the University of California at San Francisco, and Michael Drazer, a Damon Runyon physician scientist from the University of Chicago. We've asked them each to introduce themselves before we move on to our discussion. Caroline, would you like to start, please?
Sure. Yeah, I'm Caroline Bartman. I'm a postdoc in the Josh Rabinowitz lab at Princeton University. And so under normal circumstances, our lab studies the metabolism of cancer cells, and I'm specifically focused on pancreatic cancer. So the goal of that research would be to understand how the metabolism of pancreatic cancer is distinct from your healthy tissues. So ideally, you would be able to treat cancer while trying to avoid side effects on healthy tissues. And I've had the Damon Runyon Fellowship for 1 year.
Great, thank you, Caroline. Kyle, would you like to go next, please?
Yeah. Hi, everyone. My name's Kyle. I'd like to first thank Young for having us today, and thanks for everything that you and everyone at the foundation do, especially trying to support us through these difficult times. And thanks to all of the attendees who are joining. Your support means a lot to us, and anything you can do will go a long way, especially in these really uncertain times. I'm a 4th year Damon Runyon Fellow, so I've been with the foundation for a while now. And I'm a postdoc in Wendell Lim's lab. And the lab sort of focuses on understanding how cells process information. For the past about decade, the cells we've been focusing on have been immune cells. And that transition was mostly inspired by the rise of engineered T cells to fight cancer. So you may be familiar with CAR T cells, or T cells that are engineered with proteins called chimeric antigen receptors that target the T cells to a specific cancer that's chosen by the physicians or researchers that engineer the T cells. My research focuses on modifying the signals that the receptors send to the T cell to improve the ability of the immune cells to proliferate or self-replicate, kill tumors, and persist for long periods of time. So, hopefully my work will make advances that improve how T cells work in patients for both safety and the efficacy with which they clear the tumors. But my work also hopefully will kind of help push T cell engineering from kind of an ad hoc art to a more systematic endeavor.
Great, thank you. Michael?
I would like to echo what Kyle said. Thank you all for tuning in. So my name is Michael Drazer. I'm a physician scientist at the University of Chicago. And as a physician, I care for patients with blood cancers. And what I had always wondered about when I was going through my training was some patients develop these blood cancers and others obviously do not. And I've always wondered, why that is. And we now know that there are certain families where there are very clear genetic causes of their leukemia and their blood cancers. So my Damon Runyon supported research focuses on working with those families to understand why do they develop leukemia and other people do not. And the goal of that ultimately is to take that knowledge and move it to people with just kind of garden variety blood cancers and try and inform their treatments. So I've worked with Lucy Godley here, who's a pioneer in that field. And I'm in my 4th year of funding, as Kyle is as well.
Great, thank you all so much. So I would love to just start by asking you each to talk a little bit specifically about how the COVID-19 pandemic related shutdowns have impacted your day-to-day experience as a laboratory researcher. Caroline, would you like to start? Thank you.
Sure. Yeah, it's been a huge challenge. Our lab has basically been shut down for around 7 weeks. One person goes in every day just to kind of like check on things, but basically we're all working from home, which, as Young said, is a huge challenge since normally we're carrying out experiments every day. There are still some things we can do from home, like writing up papers to describe the work that we've previously completed in the lab. And then my lab in particular has made a small effort to switch a bit of our focus from cancer to looking at COVID. So I mentioned that our lab studies cancer metabolism and how cancer changes the metabolism of cells. And so now we're working with a coronavirus lab at the University of Pennsylvania to see how coronavirus infection changes the metabolism of cells. So trying to take that knowledge and the systems we've applied to cancer in the past and apply that to coronavirus infection.
And Caroline, since you mentioned that, maybe if you don't mind talking a little bit more, some of our audience may have seen that we shared a New York Times op-ed piece that Caroline wrote with her mentor, and they talked a bit about viral dose and the importance of that in understanding disease infection rate and prognosis. So I wondered if you wanted to share a little bit more about those findings.
Yeah. So when the shutdown started, my boss and I were both kind of stress reading science articles about COVID, as I imagine many of us are. And we felt that no one was raising this question of what role viral dose may play. So if you're not familiar, the idea is that in a lab where you could have a volunteer come in and give them a virus, like a very mild cold coronavirus, for example, the more viral particles you give them, the higher risk they have of catching that illness, and then the worse the disease may be. And so something we need to find out as a field is whether this is also true when people are exposed to COVID in the environment. So if that were true, that would mean that, for example, healthcare workers who are constantly maybe being exposed to virus and are exposed to the very sickest patients may be sort of at a higher risk of severe forms of the disease. So that was something that we didn't really see people discussing. And so we wrote this op-ed to try to raise this issue so that policymakers could think about it. especially when we think about how to ultimately go back to work and maybe how we could try to minimize those sorts of high viral dose infections.
Great, thank you for sharing that. And I think this highlights the point that, we often make at Damon Runyon, which is that it's incredibly important to fund basic research because we know that it has the potential to impact not only cancer, but other diseases as well. And this is a perfect example of that. Kyle, would you like to speak to how all of this has impacted your day-to-day experience?
Yeah, so the experience that I'm having and a lot of other scientists are having is very similar to what Carolyn described. So we've essentially been shut out of lab, we're working from home, and a lot of our work is, probably 80% or more of our work is really dependent on us being in lab. A lot of us have data which we, are now able to analyze from home. We're writing up papers, like Carolyn said, and also spending a lot of time reading and planning for what we'll do when labs finally reopen. And that goes beyond just the science that we'll do when we're back in. Right now, we're spending a lot of time planning for how labs will operate once they reopen. I think, like the rest of the world, things are going to be a little bit different. We'll have to have social distancing measures, safety measures. We'll be probably doing shift work, and all of that's going to make it's going to make research really difficult because a lot of the stuff that we do takes hours of attention or very specific scheduling to get done. So everything that we want to do when we go back in, we'll have to take all of these things into account and figure out how to do that in kind of a what everyone's calling the post-COVID world. So so we're spending a lot of time working on things like that, and that's kind of it. But I also want to say, I know everyone's going through a tough time, but it's pretty frustrating. But I also think everyone's really excited, and everyone wants to get back into work and work on these really tough problems.
Mm-hmm. Great, thank you. Michael, could you tell us about your experience, both on the research side as well as a clinician? How have things been going for you?
Yeah, I think it's been kind of a double hit, obviously, because I have a lot of patients who are very susceptible to COVID and have very poor outcomes, unfortunately, when they are infected. So, as Caroline and Kyle had mentioned, we have designated someone to come in every day to the lab to take care of things that just have to be done. And that person has been me, because I tend to be here anyways for my patients. So they're getting a lot of use out of me right now. But I think that's probably the safest in terms of the spirit of social distancing. But, I've had a number of patients who have gone through COVID infections, very severe life-threatening COVID infections. And so it's been very striking to see, you understand the gravity of the situation when you see what these people are going through. And many of these people already were going to be going through treatments in the hospital for 4 to 6 weeks. And they depend on their family and people like that to be there to support them. And many of those patients are all alone now for 4 to 6 weeks by themselves. So, I think it's, as Kyle mentioned, and Carolyn also mentioned, I think that at the end of this, I think people are going to have a very renewed enthusiasm in many ways for the work that we do. But I think that in the short term, it is very sobering to see what these patients are going through on a day-to-day basis. So I don't really complain when I'm coming in every day to check various, liquid nitrogen levels and things like that, to make sure that our supplies that we've developed for the years are safe, because I think that, I get a phone call probably every other hour from a patient who's concerned about COVID or a family member who's concerned about COVID.
Michael, have you had to delay treatment decisions for some of your patients as well due to the COVID situation in the hospitals?
That's a great question. I think that we have had to delay treatments. I'm thinking about a number of patients in particular where one of the treatments that we do is called stem cell transplant for blood cancers. So we take someone's stem cells from a donor and we basically donate them to a person with a blood cancer. And that's a very intensive procedure. And those people are very, very immunocompromised during that procedure. And that takes about 4 to 6 weeks. And I've had a number of patients where in a non-COVID world, they would be going through that right now. I mean, that's a curative treatment in many cases. And we've unfortunately had to delay that. In Chicago here, we've been able to get a few patients through, but the numbers are much lower than we would normally be doing. The same goes for you all in New York. I have some colleagues who I share patients with actually in New York, and they are doing the same thing. They're very much slowing down those curative procedures. So it's been tough because some of my patients, I can send them to other cities that COVID has not affected. But I work on the south side of Chicago where the poverty level is quite high. And most of my patients don't have the means to be able to travel to different kind of destinations to get that curative treatment. So we're getting very creative in the way that we're doing things for these patients and kind of hoping for the best.
Well, thank you for all that you're doing. We fund a number of physician scientists, and I know that they are all having to really increase the amount of time that they spend in the clinic, and they're stepping up and helping where they can to treat the COVID patients. But certainly this has a very significant impact on cancer patients as well. So this is a question specifically for Kyle and Michael. Both of you are scheduled to be ending your Damon Runyon Awards at the end of June, which is coming up very quickly. And so, if we know that for you, that these shutdowns are particularly disruptive for your career trajectory and your career transitions that you may have been planning. And so that's why we had made the decision to provide the extension funding for scientists like you who are in this situation. Could you both speak a little bit to what this means for this time period that's very critical, where you're thinking about transitioning to the next step in your career where you can pursue an independent position?
Right. Yeah. So obviously the shutdown has been disruptive for everyone in society, and it's been disruptive for all of the scientists that we work with. But I think it's safe to say that everyone who's in kind of a transitional period in their lives or careers has been really affected. So, if you're at the end of your high school or college career and you were going to transition, it's extremely disruptive. And it's similarly disruptive for, I think, probably the fourth year fellows. Like myself, a lot of the fellows in my year are kind of finishing up several projects. And so my main project and my side projects are all kind of, nearing completion. And so I'm right at the point where I would probably be collecting the data that I need right now to publish on my projects. And I need those publications to apply for example, jobs and transitional funding for the next stage of my career. So there's kind of this domino effect where everything that needs to kind of go forward right now is either postponed indefinitely or just left in limbo because of the timing of all of this. So the timing has been especially disruptive, I think, for the fourth year fellows. And I think what's also been disruptive is kind of, if you did want to go on the job market, which I'd like to do, a lot of institutions right now have hiring freezes. So just by the nature of their hiring freezes, even if we're in a position to apply, our entire academic and research timeline is being shifted by however long those job and hiring freezes last. So it's been particularly disruptive, and I think you don't just lose the time that you're shut out of lab. You kind of lose a little bit of time going into that and a little bit of time coming out of it. So so if we can kind of recover some of this time and make up for what we've lost, I think it'll go a long way after this.
Kyle, I have a question for you. In terms of your projects, are you going to be able to pick them up right where they were, or are they going to be set back and you have to kind of walk backwards a little bit?
Yeah, they're set back. I mean, I do a lot of research with all of the cells I use are from humans, so they're called primary human T cells, and we do in vitro work in test tubes, but we also do mouse work. So it's going to take us a bit of time to get the human T cell projects back up and running, because human T cells grow at whatever pace they'd like to, and then mice kind of are on their own. They're on their own time also. So like I said, you kind of lose time going into the shutdown. And that's the time it takes to shut things down and the time it takes that you lost, like not being able to finish experiments that were in progress. And then also the time coming out of the shutdown will be the time ramping things up. So acquiring the materials again, getting them to grow, restarting some of the mouse work.
Thanks. I think very similar to Kyle, I was in the beginning stages of my job hunt, and I completely agree with what he said about the hiring freezes, because in terms of starting a new lab, which is where people like Kyle and I are, there's a large capital investment that's required on the part of the institution where you get hired. And especially as a physician, I can make them a lot more money seeing patients all the time, which is not really why I went down this path. I see patients frequently, but the main focus here is doing the research in the lab for those patients. So what's happening is a lot of people are saying, we'll hire you if you can see patients for a year or two, and then the money will come later for your research. And I think people who really are on the ground doing research, we've been disrupted now for about 6 or 7 weeks here in Chicago. The idea of disrupting our research programs for a year or two, I mean, that would be completely crippling. So what we've been trying to do in terms of that transition that Kyle is referencing is trying to find ways to kind of cobble together support for this transitional period in particular, to basically keep the lights on and maybe do a little bit more of a bare bones research program until the economy basically recovers and these institutions feel a little bit more confident giving us the type of resources that we need, especially as early career people who are starting from scratch, essentially.
Great. Well, and how did it, how meaningful is it that Damon Runyon will be providing you with some additional support? How did you feel when you found out that we would be offering this extension funding?
I think it's incredibly meaningful. Right now, all of our advisors are, providing for us. So they're paying everyone who works in the lab, the technicians, the grad students, the postdocs. And a lot of people right now, unfortunately, don't have a means to pay the bills. And we're lucky enough that we're still getting paid, but that does come kind of at a cost to the research itself. In a worse world, our advisors would, take away our funding. We'd be without a paycheck, but that money would go to research in the future. And right now we're getting paid, but essentially that money's not getting kind of its scientific bang for the buck, right? We're not doing the work that the money's kind of paying us to do. So I think it means a lot to our advisors and the other people in the lab, because every dollar that they have to pay us, that we have to stay later, especially as fourth years, so, I'm going to be staying, and there are younger people in the lab who could be getting paid after this. And I don't want to take funding from them, but essentially that's what this kind of shutdown would be causing. That won't be happening for whatever period of time Damon Runyon chooses to extend our funding. And also, I think there's just not necessarily that we'll get fired, but money could be tight after this. We don't know exactly what the funding landscape is going to look like. I think our funding futures and scientific futures are extremely uncertain right now. And so the time that's given to us to finish projects and look for jobs will be incredibly important to making sure that we don't kind of get forced out of science.
I think I agree with everything Kyle said. And the other thing I will add on to that is I literally gave a talk before all this happened about a week before that. I said that Damon Runyon invests in people, not projects. And I had told Young and some other people during a conference call that. And then they told me about this. And I said, that encapsulates that approach. So I think that everything Kyle said, I completely endorse. And then I would add that on. That was my first impression, was this is another investment in people who have spent most times, longer than a decade pursuing this course of research, basically this career path. And to kind of give up now would be obviously a huge shame.
Yes, absolutely. Well, thank you for sharing that. For us, it would be really a tragedy for this situation to push any of our amazing scientists out of the field, and for them not to have the resources to continue this very important work would really, as you said, be very crippling. for the space. So maybe we can turn to, some other positive things, which are, I would love to hear from all of you how you've seen some examples perhaps of how the scientific community has come together during this time to work in collaboration potentially on, existing projects or new projects. Can any of you speak to that?
I mean, in a very broad sense, it's been really inspiring seeing how basic researchers, I mean, particularly epidemiologists and virologists, have been working, around the clock so hard, and we can already see the fruits of that work being published, and we're already gaining a better understanding of this virus. Basically, and that's a legacy of the investment our country and organizations like Damon Runyon have made in establishing like great universities and great research. So that's been a one, tiny silver lining to this horrible situation, I think.
Yeah, there are also some kind of non-research things that scientists have done. I know that at a number of institutions like UCSF, there are these kind of committees that do, they're involved in doing things that aren't exactly related to research. And at UCSF, they got together and they sourced a lot of materials directly from research labs that they knew would be shut down. So, they knew we were shutting down. They came around and collected as much ethanol as they could, as much hydrogen peroxide as they could, and collected other materials that they used to make, enormous amounts of hand sanitizers for the local hospitals, right as hand sanitizers were becoming difficult to acquire. So they were able to produce huge amounts and deliver them, which I think made a big difference in the response to the initial response to the disease in the Bay Area, at least.
I think that from my standpoint, a lot of the collaborations that we already had in the field oftentimes cross national boundaries and time zones, and we've had a number of collaborations that have we've basically pivoted to those a little bit more than we normally would while we're staying at home. And there's no better time to get to know some of your colleagues who you otherwise don't really get to know all that well than during a global pandemic. So we're all getting very accustomed to Zoom, and you can't hide some things on Zoom, like kids running around in the background in my case.
Great. And how have you been able to stay in touch with your colleagues in your labs and your, mentors, for example? Can you give us a sense of what that's been like for you?
Yeah, I do think, obviously not being able to do experiments is really setting us back. But I think another thing that is really hard to maintain remotely is sort of chatting about science and that scientific interchange. At Princeton in particular, we have like tea time at 4 p.m. every day, and we go and chat about science, and we discuss, how we could combine the projects we're doing or how they could inform each other. And that sort of just talking about science and thinking about science is so valuable in coming up with things that might not have occurred to you otherwise. And so our lab is trying to do, we have a weekly meeting with the whole group, and then we also have smaller sort of breakout sessions of people with related projects. And I think we're trying to sort of fill the void with maintaining those interactions.
Like Carolyn's lab, my lab meets once a week. We have our weekly group meeting by Zoom instead of in a normal meeting room. I have a weekly one-on-one with my advisor that, again, instead of being in person, is by Zoom. And as soon as the shutdown started, my entire lab adopted Slack, the communication platform. So we're constantly messaging over Slack so we can post papers that we've read. A lot of people in the lab who are finishing up papers post their papers for others to review. And we've also been using Slack to kind of communicate about how we're going to consolidate projects when we start, because we'll have to work in teams and shifts. We won't be able to finish our projects the way that we normally would. So we've been trying to kind of work out exactly how this new teamwork and structure is going to look once we're back in lab.
I think for our group, we have a lot of various skill sets in the group. So we have some people who are more kind of bioinformatics heavy. They primarily do programming and that type of thing. And then there are people who are more involved in the wet lab, doing experiments with test tubes, et cetera. And this has been a time where people have really pivoted to learn new techniques from other people in the lab. And I think that's been very refreshing to see that we have some of the youngest members of our lab, not surprisingly, tend to be the most savvy with programming. So the youngest members of our lab are teaching the elders. So it's been very, very fun to see that as people kind of shine in their own way.
That's great. I know that Carolyn, you mentioned, at the beginning that the stress reading of COVID-related papers that I think many of us are doing. I'm sure I know that people within our audience are definitely interested in getting your perspective as scientists about what areas of COVID-related research you think are most promising. So I wonder if any of you have some comments about that.
Something I think is in the long term, I'm really hopeful about is vaccine development. We've seen, I think there are like 10 or 12 different vaccines with different strategies in different countries. And I'm really optimistic that, at least some of those will be helpful in the sort of medium term, like whatever, 1 to 3 years, in really helping to tame the pandemic. Yeah.
I'm a big fan of immunotherapy. I mean, it's the space I work in. And so I've been kind of following what's going on with the monoclonal antibodies that have, they're used for other diseases right now, but what they really do is they block targets. And so there is a monoclonal antibody that blocks a protein called the IL-6 receptor, and IL-6 is involved in, modulating things like inflammation. And there's a real problem that comes with COVID-19 is cytokine release syndrome. And that's, I think, a major driver of the mortality. So I think, one of the critical things for lowering the mortality for COVID-19 is going to be, lowering cytokine release syndrome. So I think, if the IL-6 receptor blocker doesn't work, I think there's a good chance that another immune receptor blocker will help control the cytokine release syndrome. And it's a symptom that's important for COVID-19. It's also important in a lot of cancer treatments. And I think there's a lot of space out there to fix the cytokine release syndrome. So I'm really excited about that and to see where that goes.
From my perspective, whenever I see, as a clinician, I put my doctor's hat on it. When I see people having very variable responses to COVID infection, some people, we've all heard of these people who have relatively minor symptoms, and then we have other people who obviously are unfortunately passing away from this disease. And Caroline kind of keyed in on this on her editorial that she wrote for the New York Times, that maybe there's some component from the infection itself, right? The quality of the infection, the dose of the virus that people are being exposed to. I think the other part of that is we all genetically are individual people, right? And when I see variability in a response either to a chemotherapy drug or to an infection like this, that immediately gets my scientist hat and the wheels of my scientist brain spinning, right? And I say, I wonder genetically what potentially might be contributing to these variable responses that people are seeing either to treatment or the infection itself. And I think that appropriately, people are focusing on the therapeutics right now. I think obviously there's the biggest need for that. But in the long term, I think that these infections and these outbreaks are going to be happening more frequently. And I think that having a better understanding of who is truly at risk, at the greatest risk on a genetic level, will be informative for basically identifying high-risk people who, if they have some type of genetic background that makes them especially susceptible to a lethal COVID infection, they should immediately be pulled out of the workforce, right? Where some other people might be able to tolerate some of these infections. So I think that we have to think about a post-COVID world and identify people who are more resistant to these infections and really lean on those people. But that will be in the second wave of research, I think.
Mm-hmm. Great. So, Michael, we actually have a question from the audience that I think is most specifically for you. So the question relates to how you make decisions regarding whether to give a cancer patient treatment that weakens their immune system and potentially makes them more vulnerable to COVID-19.
Yeah.
I know there's probably no perfect answer for that.
Yeah, so that's a great question from Ms. Rosenfield. So thanks for asking. I think the short answer is it's completely individualized, and a lot depends on the type of cancer that we're treating. So some of these cancers, an acute leukemia, that's essentially a medical emergency, and those people need to be brought into the hospital right away. And referencing the patient I had mentioned earlier, who has been alone for 6 weeks by themselves in the hospital, that's what these people are going through. Unfortunately, they come into the hospital for a curative treatment, but they have to be there for 6 weeks, and we just have to move forward because otherwise they won't survive. And our nurses on the leukemia floor are exceptionally talented people, and they have really done a fantastic job as the true frontline workers here, and caring for these people in as safe as a manner as they possibly can. There are other people who maybe their treatments are a little bit more kind of established at this point, and we're kind of moving slowly along a defined treatment paradigm. And those are truly individual decisions. We don't have any evidence to say if we delay treatment right now, what will happen. So we have to weigh the pros and the cons with the family in the room, or via video generally now. And we have to basically make a hard decision about what we want to do. And so far, we've had, knock on wood, everyone's done well with that approach. But we're definitely getting very creative. And the newest thing, and I would encourage anyone on the call who's still listening, the newest thing that we're running into is our blood shortages and platelet shortages. So my patients, when they go through these treatments, we basically damage their bone marrow to the point that they can't make their own blood and their own platelets, which keep us from bleeding to death. And unfortunately, the donor pool has completely evaporated during this time period. And for the first time this week, we've had to ration our transfusions for our patients. So I think this is the newest thing that's happening right now. So I'd encourage people, if they're able to donate blood or platelets, they will get to the people who need them one way or the other. We have a system in the United States for basically getting these wherever they need to go. But that's something that materially, if people are asking, well, what can I do on a personal level? That's absolutely something that would directly benefit some person with cancer right now.
Great. So I wanted to, turn back to Damon Runyon in the final minutes that we have here on our webinar, and ask each of you to talk a little bit about how the Damon Runyon support and this award has shaped your career trajectory so far. What's the impact that it's had to be supported by an organization like ours?
I mean, it's been really amazing. Having the, resources that Damon Runyon gives you allows you to make choices sort of a little bit more free of the cost of experiments, and instead try to focus on the best experiment to do at that moment. So I think in terms of empowering good science, it's a really important thing for my work, and I guess all of our work. And then beyond that, it's been really exciting. So there are retreats every 2 years where all the fellows meet each other, and that was amazing. It was great to meet the other people and hear about the amazing work that they're doing to try to understand and treat cancer better. And then an important thing beyond the money itself is that Damon Runyon is very respected in the research community, and just the honor of having that accolade is super important for our careers going forward. So it's a huge honor to be funded by Damon Runyon.
I would echo what Carolyn said about the retreats, which, I love the retreats. They're amazing for meeting other scientists, talking about science, sharing your own And I also think the reputation of Damon Runyon is extremely important. If it's on your CV, it's definitely going to help. But also, I think the funding, particularly that it's 4 years. I think when I applied, it was 3 years, and you guys transitioned to 4 years that same application cycle or somewhere around them. And that extra year made a huge difference. I know that myself and several other people accepted Damon Runyon over, for example, like the Jane Coffin Childs Memorial Fund, which is another prestigious fellowship, because it was 4 years, and an extra year funding is, just incredible. It's super critical, especially if you're working on, very difficult problems that take a long time to answer. And so that the time that the fellowship has provided has just been essential to finishing a really difficult project.
I agree with all those things. I think I tell people that the funding is really transformative for physician scientists because we especially really require a lot of incubation. We basically walk away from science to do our medical training, right? For 4 years of medical school, 3 years of residency, 3 years of fellowship in my case. And, my entire field that I work in, techniques came and went in that field in between my last scientific training and what I'm doing now. So there's kind of a catch-up period that you have to do. And during the same time, the people who are pulling the strings, they say, can you see some extra patients? Can you do this? Can you do that? And there's a lot of distractions. And in my experience, when I say, I just point to the Damon Runyon, I say, this tells me that I can't do those things. And they respect that. And they know how meaningful of a award it is. And it makes them look good too, for people to have it, frankly. So that has really protected me from some of these distractions that really will ruin that incubation of a physician scientist.
Great, thank you for sharing all of that. So we hope that you will all be able to return to your labs very soon. Obviously, there's a lot of uncertainty in the world right now, and so we don't know when that will be. I want to close with one final question from our audience, which relates to whether there are any discussions that you're hearing from your institutions regarding a plan to help you return to research at full capacity. Are there any centralized, efforts that you're hearing about at this point that will help to basically put in the framework and infrastructure to enable you to get back up to speed as soon as possible?
Yeah, I think Kyle kind of referenced the fact that a lot of these institutions do have committees that include scientists and administrators for trying to best plan the way to safely get back to work. But my impression is, a lot of the social distancing things that we've been talking about in general will be really critical for going back to research. So staying 6 feet away, face masks, lots of cleaning of surfaces, and most likely working in shifts will be really ways that we try to get a fair amount done while still stopping the spread of the pandemic.
Yeah, so also going into the shutdown, there was a PI, I don't remember his name, but he was at a major university, and he'd come up with a very detailed sort of checklist for what you need to consider going into the shutdown. And I think that there are PIs who are doing kind of the same thing, thinking about coming out of the shutdown. And these lists travel, scientists are very, very interconnected. And so these lists travel very quickly from institution to institution. And also there are some kind of opinion type papers that have been coming out about some of the things we can do. There's this idea, for example, that we might do some kind of timing of how long we work in the lab based on how long it would take if you become sick and are asymptomatic. How long would it take from the infection until you're contagious? And maybe we can kind of time how long people work in lab with that time window, and then they have to be out of lab for some time window after that to make sure that, so it's kind of like coming out of isolation and going back into isolation at periods that might sort of protect people. So there are kind of ideas like that are circulating.
I would echo all the things that you both said. I think that the way that we think about pandemics is they happen very quickly, but they resolve very, very slowly. And I think that we're all in this for the long haul. And when I think about the timeline of reopening things to full strength, I mean, there are people in our lab, frankly, who I don't think they're going to come back to our lab before they move on to their next position. And that's been hard to tell some of our younger group members that, you might not be in our lab again until you come back to visit us, but you will be going to graduate school, you'll be starting your postdoctoral training somewhere. And that's been hard to see that for those people because their projects will never have a chance to be finished by them. So there's a sense of finality when you finish a project, as you all know. And there's a sense of reward and, kind of accomplishment. And those people are going to be robbed of that, which makes me sad for them.
Well, we are just about out of time, so I'd like to wrap up with a few final remarks. And first of all, just want to thank our scientists, Caroline, Kyle, and Michael. Thank you so much for joining us today and for sharing your knowledge and experiences with us. And again, for continuing to do what you do and to inspire us all with your commitment and dedication and resilience during this very challenging time. Thanks to all of you who are in our audience for joining us today. And as we navigate this next chapter as a community, I'm incredibly grateful to know that we're moving forward with all of you as our partners. Your continued generous support of Damon Runyon will ensure that we can remain focused on our mission of providing our brave and bold scientists with the resources necessary to make future breakthroughs against cancer. We know that this is the time when many of you may be looking for opportunities to help solve problems and make an immediate impact through your philanthropy. And we hope that you'll consider joining our efforts to make a meaningful difference by ensuring that our scientists on the verge of new discoveries don't lose any more precious time once it is safe for them to restart their research programs again. So I look forward to talking to all of you and seeing you soon, hopefully in person. Thank you again for joining us, and from all of us here at Damon Runyon, take care, stay safe, and be well. Thank you.
Thank you.
Thanks.
Thank you, everyone.
Thank you.